Today, the topic we discuss is about transport of ammonia to the liver. So, we start the topic. Protein turnover and amino acid degradation will be going on in all tissues as a part of normal cellular metabolism and as a part of normal cellular tissue turnover. When protein is undergoing turnover and amino acids are released because of the breakdown of proteins. These amino acids are entering into the cytoplasm to get into translation process for to make a new amino acids in a cell. Another sum of the amino acid that need to be degraded, so they need to be transport it to the liver, especially the alpha amino group, that is the primary amino acid, need to be transported to the liver. Because the carbon skeleton of amino acids may into metabolic intermediate either in glycolysis or a tissue cycle to be utilized by a particular tissue, whereas the primary amino group that need to be transported to the liver for urea cycle, so that ammonia from the primary amino group need to be handled properly, otherwise it can lead to hyperammonemia, and excess ammonia is toxic to the neurons. So in this process of transport of amino group, nitrogen, which will be released as ammonia or ammonium from the peripheral tissues to the liver, need to be handled properly. That is usually done by two amino acids. One of that is alanine and the other is glutamine. Alanine and glutamine are the major carriers of nitrogen in the blood. Role of alanine in transport of ammonia from the peripheral tissues to the liver. Alanine is the primarily transporter of amino group from skeletal muscle to the liver. Predominantly, the amino acid that are released by protein turnover in the skeletal muscle in one or the other way, they will be undergoing a transamination reactions ultimately to make alanine and glutamine. So both alanine and glutamine, they are formed in the peripheral cell, especially in the skeletal muscle. So transport that alanine and glutamine out of the skeletal muscle, so that liver takes that up and break that alanine and glutamine, release amino group nitrogen and convert that into urea. So out of these two amino acids coming out of skeletal muscle, that is alanine and glutamine. Alanine is the predominant amino acid that carries amino group nitrogen from skeletal muscle. So now the transport of amino acid, carbons and nitrogen between muscle and liver is known as the glucose alanine cycle. Let's understand what exactly is the glucose alanine cycle. Amino acids which are coming out of skeletal muscle proteolysis, they will undergo transamination reaction with alpha ketoglutarate to make glutamate. That means the amino group nitrogen, the primary amino group, it will be now given to alpha ketoglutarate, which is an acceptor of that primary amino group and is now present in the glutamate, because alpha ketoglutarate is converted to glutamate while the amino acid is converted to its corresponding alpha keto acids, and that corresponding alpha keto acid will undergo its metabolic fate in the skeletal muscle. Now, the glutamate, which contains amino group from the amino acid from skeletal muscle, and that glutamate undergoes transamination reaction with the pyruvate. You know, pyruvate is the glycolytic intermediate. By the way, it accepts the amino group from glutamate and becomes alanine. So your amino group is now in alanine. So basically it comes from amino acid to glutamate and glutamate to new alanine. Now the alanine is the predominant type of amino acid that gets out of the skeletal muscle in the circulation, and in the circulation it will when they reach liver, and liver is going to take that alanine into the hepatocytes, and alanine in the hepatocytes are undergoing transamination reaction with alpha ketoglutarate, where it will give its amino group to alpha ketoglutarate to become glutamate, and alanine itself is now converted to pyruvate because it has donated its amino group to alpha ketoglutarate, and alpha ketoglutarate is converted to glutamine. So what will happen to the pyruvate? Pyruvate can be getting into acetyl CoA formation and tissue cycle, or if is in fasting condition, if there is a need for glucose, so pyruvate is converted into glucose, and glucose enters into the blood, and that glucose usually taken up by the RBCs and brain during fasting and starvation. Now, what about the amino group that came from all the way from skeletal muscles in the form of alanine, and in the liver that alanine amino group is given to the glutamate? Now the glutamate will undergo glutamate dehydrogenase reaction and release that amino group, and that amino group will become a part of urea, and this is how ammonia, basically the NH3 there is coming from the primary amino group, is transported to the liver from for urea cycle. That's how ammonia is neutralized in the liver by making it urea, which is less toxic molecule. Otherwise it's ammonia and ammonium ion that is toxic to central nervous system. Role of glutamine in transport of ammonia to the liver. Other than skeletal muscles, where alanine is the predominant amino acid that is getting in out of the skeletal muscle to transport amino group nitrogen to the liver, even the skeletal muscle secretes the glutamine as a transporter of amino group nitrogen to the liver, but predominant one is alanine, whereas all other peripheral tissues they will transport the amino group nitrogen out of the tissue into the blood in the form of glutamine. So peripheral tissues have two enzymes. Whenever they will higher ammonia level, they can fix ammonia to an alpha ketoglutarate to make glutamate, and glutamate can take up one more ammonium ion to become glutamine. The two enzymes are glutamate dehydrogenase and glutamine synthetase. In the liver, glutaminase, glutamate dehydrogenase, glutamine is taken up by the liver, kidney, and intestine. Liver makes urea. Kidney is second line of defense for excess ammonia in the kidney, and intestine uses glutamine as fuel. In the muscles and peripheral tissues, so whenever there is higher levels of ammonium ion, so ammonium ion will be fixed with alpha ketoglutarate to make glutamate. So alpha ketoglutarate accepts an ammonium ion and be converted to glutamate, and that will be done by enzyme glutamate dehydrogenase. Fixation of ammonium and alpha ketoglutarate done by glutamate dehydrogenase enzyme, and it uses NADPH. Now the glutamate will be fixed with one more ammonium ion done by glutamine synthetase enzyme, which uses ATP to do that and become glutamine, and that is how glutamine is made in the peripheral tissues including muscles. Now the glutamine will be secreted out into the blood, and that will be taken up by the urea, and in the liver, especially in the mitochondria. These are all mitochondrial reactions. Glutamine is deaminated into glutamate by glutaminase enzyme, and that ammonium ion which is going out here will be getting into urea cycle, and that's how it will be neutralized. And now what happened to the glutamate? Glutamate undergoes glutamate dehydrogenase reaction in mitochondria of the liver, release one ammonium ion, and now these two ammonium ions will get into urea cycle, and urea formation is going on in the liver, and urea will be secreted out in kidney, going to filter the urea out into the urine, and that's how urea will get out of our body, carrying two ammonium ion through the kidney into the urine. So that how our transport of ammonium ion goes from peripheral tissues to the liver, and in the liver it will be incorporated into the urea formation.